You gotta let me know, are we human? Or my sign is vital, my hands are cold, and I'm all I need, looking for the answer. Are we human? Or are we dancers? Are we human? Or are we dancers? Are we human? Or are we dancers? On the corner of Main Street, just trying to keep it in line. Say you wanna move on, and you say I'm falling behind. Can you read my mind? You read my mind. Never really gave up on breaking out of this two-star town. I got the green light, I got a little fight. Gonna turn this thing around. Can you read my mind? Read my mind. Good old days, the honest man, restless heart, the promised land, the subtle kiss that no one sees, a broken wrist and a picture piece of what I don't mind. You don't mind, 'cause I don't shine. You don't shine before you go. When you read my mind. Funny how you just break down, waiting on some sign. I pull up to the front of your driveway, magic soaking my spine. You read my mind. You read my mind. Teenage queen, the long gun, the drop dead dream, the chosen one, southern draw, the world, see, city one, trembling away. I don't mind, you don't mind, 'cause I don't shine, you don't shine before you jump. Tell me what you found. When you read my mind. She said, I don't mind if you don't mind, 'cause I don't shine, shine, put your back, I'll put you back on me. Stars are blazing like rebel diamonds cut out of the sun. Can you read my mind? Make some face. You know you've only got one. Change your ways while you're young. Girl, you understand why I like you. Smile like you. Looking back at the sunsets on the side, we lost track of the time. Dreams are like they used to be. Something so carelessly. Smile like you. That's almost that's almost that's almost now the same streets that I did, all the same streets that I did. That's why I like you made smile like you. Smile like you. Great, take my hand and come with me because you look so fine that I really want to make you mine. You look so fine that I really want to make you mine. 4, 5, 6, come on and get your kicks. Now you don't need the money when you look like that, do you, honey? A big black room, a long brown hair. She's so sweet with her. Damn. Well, I can say you are with me. About you with that. No way. Much the same. I'm never gonna let you go. I said, are you gonna be my girl? Take my hand and come with me because you look so fine that I really want to make you mine. So fine that I really want to make you mine. 5, 6, come on and get your kicks. Now you don't need money with a face like that, do ya? Long brown hair. She's so sweet with her. Yeah. Well, I can say, you was me. About you, listen. No, wait. What's the sound? I said, are you gonna be my girl?
Good morning, everybody, and welcome to our second session of our symposium on overcoming challenges in biodiversity modelling. We had some wonderful talks yesterday, and I'm sure you're going to hear some more wonderful talks today. My name's Natalie Briscoe, and I'll be running the session today. So let's get straight into it because we've got a lot of talks to get through. Some quick housekeeping. The translation code is just outside the front door if anyone needs that one. So our first presenter will be Abbie Jones, and she'll be presenting on biases overpowering biodiversity scale relationships, insights from 10,000 plant species.
Hi, everyone. So I'm Abbie Jones. I'm a PhD student in the lab of Laura Pollock at McGill University. And today I'm going to be presenting to you some works that I've done as part of my, my PhD thesis. Nope, that way. Nope. There we go. All right, so let's start big picture. So land, the availability of land around us is something that we often take for granted, maybe less so in this audience here, but as a society in general. However, land is a finite resource, of course, and we must learn to develop and coexist with our surroundings and its nature. Particularly in highly developed areas such as Europe, it's been quite a strong recent movement to kind of restore and start protecting more and more, or creating more and more protected area by the day. And this is where the Natura 2000 protected area network came into play. So in 2000, the European Union selected 18% of its territories, both marine and terrestrial, for various levels of protection. And now, as part of the growing 30 by 30 movement, the European Union is committed to protecting 30% of its lands. So they already have 18% as of 2000, but continuing on to 30% of its lands by the year 2030. However, this begs the question, how exactly are lands protected and selected for protection? Because in general, reliable knowledge of the distribution of Earth's biodiversity is crucial for effective nature policy. However, few species have data rich enough to kind of interpret their entire distributions, and let alone for entire species assemblages. So how are we to know how species are distributed along a landscape, and what are the quantitative tools that we can use to generate such estimates? And that is where species distribution models, or SDMs, often come into play. So they are used to estimate species ranges, joining species locations data, such as occurrences on GBIF, with environmental drivers in order to generate predictions as to where species are likely to be. When you use them for many species, you can then generate biodiversity estimates or just species richness estimates by simply summing your probabilities or your predictions. However, traditional SDMs, or SDMs fit using traditional methods, often have a high rates of imperfect detections, often due to high levels of sampling biases, where you can end up like hindering conservation efforts or just simply clouding our knowledge of the patterns of this biodiversity. So what are these sampling biases, right? Well, Like I said before, for many SDMs, the data that we use are the species data that we use, pardon me, is generated or obtained from GBIF or other citizen science databases. Due to the stochastic nature of this data, the data does not simply represent where the species is, but also where the people tend to be and where the people tend to sample it. So these biases exist both at a continental level, where data tends to pool in countries with higher levels of research or higher levels of GDP, but also locally within a given country or region, where data is going to pool around zones of high population density or around roads. These biases are then going to trickle into your SDM outputs unless they're appropriately taken into account. Once we have all the estimates of species distributions that we want, when it comes time to plan a new protected area, spatial prioritization tools are often used. So these are prioritization algorithms that can take in a multitude of different priorities, such as a bunch of different species distributions, and provide optimized suggestions of land protections which maximize different species coverage. We know that decisions pertaining to SDMs fitting do make their way into spatial prioritization. So a recent study investigated this by building SDMs with different modeling decisions and compared their spatial priority ranks. And all decisions involved in the modeling process did impact the priority ranks, but most notably was the bias adjustments. So whether or not you accounted for bias in your models, that most impacted where you end up spatially prioritizing. Then there comes the additional question of the scale of our predictions in our modeling. So due to differing patterns of alpha diversity and species turnover, the species diversity predictions that we generate may be totally different depending on the scale at which we build our models. So we need both reliable outputs that are fine scale, let's say to 1 kilometer, which is usually most useful for stakeholders, but also broad scale patterns that can help us understand the patterns of entire biogeographical species pools. And furthermore, we specifically need to understand how sampling biases trickle into SDM outputs differently at different scales, which has not yet been done to a very large extent. So this is where our set of papers come in. So here we wanted to compare the impacts that various methods of bias-corrected SDMs may have on, first, the modeled biodiversity, so our outputs of our SDMs, and then second, on spatial prioritization. And through this, and by identifying best SDM practices for highly biased landscapes, we wanted to help avoid the wasting of limited land conservation resources. So we selected Europe as our study system just due to the sheer volume of the data that is available, the species data, both in terms of occurrences, so presence-only data, citizen science data, which we've heard a lot about at this conference, but also in the format of presence-absence data or survey-type data, which tend to be more scientifically collected and rigorous. However, while very rich overall, the European data landscape is still very polarized in terms of its locational biases, where there are strong dichotomies between the western parts of the continent, which tend to be quite data rich, and the eastern parts of the continent, which can be quite lacking. We had a total of 10,000 modelable species, and our goal was to compare commonly used modeling methods that both do and don't take sampling bias into account in their methodology. So the first modeling decision that we introduced was, like I've mentioned before, the data input types. So you can fit SDMs either using presence-only data or presence-absence data. And so we wanted to compare the two. But in addition to those two, we also generated a very simple integrated model that incorporated both data types into a single algorithm as a first potential way to account for bias. We then introduced 3 additional model variants, so modeling decisions in our study design, so algorithm choice, for which we had 3, and 2 bias control methods, so the pseudo-absence selection or the background points, as well as the predictors included. Once all of our models were fit and validated, I used the software Zonation 5 in order to spatially prioritize the area and also generate the layers of endemism and species richness. To assess for differences between prioritization scenarios, I first ranked or extracted the top priority ranks of the top 30%, the top 1%, the top 5%, and the top 17% of cells across the landscape, and I compared that selection quantitatively. And importantly, we performed all of our analyses at 3 different scales. So at the 1 by 1 kilometer level for our more fine scale alpha diversity estimates, all the way to the 100 by 100 kilometer level in order for us to get an idea of these broad scale patterns of biodiversity. Moving on to results, already when just plotting our species data, we are kind of seeing the root of the bias issue when it comes to the occurrences, which you can see on top. So there's a hotbed of occurrence records in France, Belgium, the Netherlands, Belgium, the Netherlands, Germany, and the tip of southern Sweden, where this area in Europe contains more occurrences than the rest of Europe combined by far. Are these Northwestern Europe occurrences still controllable by traditional bias control methods, or is bias still going to trickle into our SDM outputs? In short, the answer is yes, the bias are still going to trickle into our SDM outputs. So plotted here, you are seeing the species richness, so our SDM outputs all summed together for both bias-corrected presence-only models, which you can see on the left, and presence-absence models that you can see on the right. And I want to emphasize again that on the left, for the presence-only models, these are the bias-corrected models. So first, by far, we found that the input data was the most important variable when it came to impacting the differences that we observed within our patterns. But most alarmingly, we found that the corrected presence-only models still systematically and overemphasized data-rich and population-dense areas at the 1 by 1 kilometer level, leading to strong inflation of species richness in certain areas. At all scales, sampling biases for presence-only data distorted our estimates of biodiversity, and it did so in unequal ways across biogeographical regions, which ended up overpowering some known biogeographical patterns. So for example, the Alps and other alpine regions of Europe are known hotspots of biodiversity due to their high levels of species turnover, habitat heterogeneity, and endemism. However, in our presence-only models, in comparison to that Northwestern emphasis, the Alps come up as a complete cold spot. And if you compare that to our more rigorously collected presence-absence models, which did show up the Alps, this Northwestern emphasis is consistent, albeit lessened at different scales as we increase the scales. However, we also found that at the 1 kilometer scale, presence-absence models, on the other hand, failed to highlight hotspots in the Mediterranean. So the Mediterranean is another global hotspot of biodiversity, generally recognized to be so due to its high levels of endemism, where 80% of European endemics are Mediterranean. If the Mediterranean is so species rich, why are we not seeing it lit up at this scale here? Well, after scaling up our models, we believe that since the Mediterranean's biodiversity comes through its endemism and rates of species turnover, not through its alpha diversity, these become visible at larger scales as more species pools are agglomerated together. So at larger scales, the Mediterranean had hotspots that were only paralleled by the Alps. Therefore, when we're assessing these regional patterns of biodiversity, the scale at which you model is vital, as you otherwise might be missing out on an entirely rich species pool. So we now know that our different modeling decisions do impact the biodiversity patterns that we observe. However, how do these decisions then trickle into spatial prioritization? And do we still observe large differences, or do the model scenarios kind of converge? Well, here we have different maps showing the scenario convergence of various modeling decisions that I've mentioned, meaning how well do these different modeling scenarios agree on what the 30% of lands they have identified is. So while we found that there is some convergence of scenarios, mostly along the Alps and the Mediterranean, which makes sense as these spatial prioritization algorithms typically highlight zones of high endemism, they don't agree on where, and they don't agree on how important these hotspots are. So convergence was really only present for the overall 30% of lands. When we fall into the nitty-gritty, into the fine scale priorities, like identifying the top 1%, the top 5%, the models had instead very low levels of agreement. So decisions pertaining to bias control matter much more the more specific your priorities are. After further breaking down our scenarios quantitatively, by far the biggest driver of differences was still the input data that we added into the models, where presence absence and presence only models were the least correlated. However, if we go into only the presence only models, only the models built on occurrences, and break down those patterns, it was pseudo absence selection that drove the biggest difference amongst these. So after reducing all scenarios into NMDS space, sort of like a PCA, pseudo absence selection had a greatest role on the first axis of variation, which accounts for the most variation. Algorithm selection was second, while the addition of covariate bias variables did not do much of anything. So to recap, pervasive sampling biases trickle into our predictions, our SDM predictions, regardless of the scale or the bias control approach that we've done. And it does so to an extent where sampling biases are overpowering some expected biogeographic patterns of biodiversity, and it impacts the prioritization scenarios severely. We believe that traditional bias control methods are only making a dent into the sampling biases that exist worldwide. And if such biases are present on models fit on the overall European landscape, which is amongst the world's most sampled continents, they can be expected to have an even stronger role in continents that are lacking much more data. This highlights the vital need for sampling bias critical approaches to biodiversity predictions, particularly instead of those that use single non-comparative approach that are then used to pinpoint important areas for conservation. All said, we must just recognize that the simple bias adjustments that we use will only be as good as the data we use to fit them. And with that, I'd like to thank everybody in my lab, so the Quantitative Biodiversity Lab, led by Laura Pollock. And if we have time, I can take any questions.
Thanks, Abbie. We have a time for one very quick question. Justin?
Thanks. That was really interesting. I think it's a really important point. I just wondered, when we're coming up with, say, 30% of the land that we want to protect, we want to have land that's going to be valuable in the future too, not just now. So how does this thinking apply to projecting where most species would be in the future and where we want to have climate resilient landscapes protected?
That's very interesting, and kind of where my future steps that I'm thinking about have often ended up, in that I'm interested in also figuring out exactly how this projects using like different, either different land use scenarios, which are expected to vary severely, like in Europe, their precipitation patterns are expected to completely shift around. So yeah, very good question, and definitely something that I'm thinking about. I'm not sure what's hard about that is, how do we quantify the impacts that we expect these biases to have in the future? Are the biases going to stay the same? Are they not? Are they not going to stay the same as our urban areas potentially shift? So I think there's a lot to think about there. Yeah.
Thanks, Abbie. So next up, we have Jakob Nyström, who will be talking about, can we predict biodiversity across large geographic and taxonomic scales? Thanks, Jake.
Hello. Great. We have slides. All right. Hi, everyone. My name is Jakob Nyström. I'm part of the Biodiversity Data Lab at Uppsala University in Sweden. And I mean, I'm really looking forward to this session. Yesterday was really great. We saw some really cutting edge stuff, interesting deep learning frameworks. Today, we're going to get maybe a little bit more basic. And the question we asked ourselves was, can we really predict biodiversity across very large geographic and taxonomic scales using commonly used methods and data? And if you need to leave now, I think the short answer is not really, but we'll see in the results. So this will come as no news to this audience that there's really high demand for global data products for biodiversity monitoring for impact assessment. So in this slide, we see an intactness map from the Biodiversity Intactness Index as an example. And this can be used for several use cases, like measuring the state of biodiversity in a consistent manner, monitoring goals and targets related to the GBF, and also quantifying risks and impacts in the corporate sector. So here we focused on models that underpin so-called intactness indicators, biodiversity intactness. So in this example here, we have two sampling sites where A represents some primary used vegetation, so a reference site, whereas B is a cropland site. And if we do an inventory here, we can look at what species are present, what their abundances are, and based on this, we can calculate things like the relative abundance between these two sites, the compositional similarity. So we're trying to understand particularly how human pressures impact relative biodiversity levels, and you make predictions based on that. So in order to produce global indicators or really any kind of indicator, predictions are really essential, as many people in this room will know. So it's really useful. We can use it to fill data gaps, to produce actionable continuous output maps. We can use it to assess current state, explain trends in the past, make projections for the future, but also to test the generality of ecological mechanisms. However, what we saw is that the predictive accuracy of the models that underpin some of these big, widely used global indicators have not really been well tested. So we saw that was a pretty big gap, and we wanted to see, what that actually looked like. So to do that, we used Predict, which is a database that collates data from many underlying source studies. I'm sure some people in this room will be familiar with it. It's widely used in indicators and to study human impacts on biodiversity. So it has that focus of anthropogenic pressures. So in our data, we had almost 26,000 sampling sites from almost 700 studies. Based on this, we used geometric mean abundance as our alpha diversity metric, the break-her dissimilarity as our beta diversity metric. This might sound like a lot of data, but on the other hand, this is on a global scale, so it's not that much. And as we've heard in many talks during this conference, there are also geographic, taxonomic gaps and biases, as to be expected in any global biodiversity data set. And another problem that we found is that a lot of these underlying source studies are very small, which creates some issues. So we can see here on these maps the number of studies and the number of sampling sites in different countries. And you can see that it's a really big spread. And in many countries, we actually have quite little data. So we didn't use Predict because it's a perfect data set. We used it as an illustrative example. It's widely used and kind of illustrates some of the issues in many global biodiversity data sets. And then, so we had Predicts, and then we wanted to test two quite common but structurally different models and apply it to this data. So first, we looked at linear mixed models. These are very common in biodiversity metasynthesis. They're used in indicators like the BII, GloBio, potentially others. And here, the focus is really on the anthropogenic pressures. So the covariates include land use, human population, road network, etc. And a lot of the sort of residual variation in the data is captured by these study level so-called random effects, which means that only the fixed effects can be used when making out-of-sample predictions. And we'll see that this turns out to be a quite big issue. And we contrasted this with a Bayesian hierarchical model, so also a hierarchical model, but where the model structure was based on biogeographic and taxonomic attributes. We also included a more holistic set of biodiversity drivers, so things like bioclimate, topography, and so on. And the key difference here is really, in my mind, how we view sort of the data generating process. So in the linear mixed model with the study level effects, we really think about the data generate process as how did we sample it? How did we collect it? Well, we collected it from these studies that sampled sort of spatial blocks. We have these random effects. Whereas in the Bayesian model, we look at it more from a sort of general point of view, looking at biogeography, taxonomy, a more holistic view on the data generating process. Okay, so how did we evaluate this? So this turned out to be a bit of a challenge given the scope of the data. And this might be why sort of it hasn't really been done for these types of models before on this data. So we used two cross-validation approaches. But the problem with all of these underlying source studies is that they are very different in their geographic, taxonomic scope, sampling methodology, and so on. So we kind of tested two cases. So one, we call that the interpolation case, where we used, I call it standard or random cross-validation, where we just randomly sample among all the sites using stratification, but you can still have sites from one study, both in the train and the test set. So it's kind of interpolating within known context. But to really stress test the models, we also use something called cross-study validation, which I think originally comes from medical research. So you essentially train on one subset of your studies and use that to predict on other studies. So this is quite hard, and this represents the extrapolation case. And given what we know about the gaps and the biases in global biodiversity data, when we actually apply these types of top-down models, we will be doing a lot of extrapolation. So this is quite interesting to see how the models are doing. So in terms of results, so there are pretty clear limits to prediction, both when it comes to interpolation and extrapolation. So here we see first the results for the alpha diversity models. So the linear mixed model in green, the Bayesian model in orange. In training, they do quite similar. In terms of standard cross-validation, so interpolation, already there, the linear mixed model does really badly, which we'll see why in the next slide. The Bayesian model does a bit better. It's not that great, so it's still really hard, but it does better. But what's really interesting is that when we do the cross-study validations, when we try to extrapolate, the performance is consistently bad, which we'll also see why. And for beta diversity, the patterns are quite similar. Overall, the performance is a bit lower, but sort of the relative differences are quite similar. And then we also hypothesized that, maybe given the design of these linear mixed models, they would actually be better at predicting differences within a study, since we are looking at the human pressures, like one site relative to another. But actually, it turns out that both models do worse when we try to predict the differences in alpha diversity levels between sites within the same study. And when we compare only to the reference sites, which is sort of at the heart of biodiversity intactness estimation, it's actually even worse. So models are not doing great. Now, the question is why? So for the linear mixed models, the core problem is that the observed variables, so the fixed effects, they describe a very small portion of the overall variation in the data, which means it's essentially impossible to do out-of-sample prediction and expect any good results. So you can see here in the second scatter plot that we get a very flat sort of predictions because we only account for the human pressures. For the Bayesian model, we looked at the distribution shifts between train and test data. So those distribution shifts are what explain the failed extrapolation performance. So if we start by looking at standard cross-validation, so I mean, in the scatter plot, we see that the model is, not great. It's not perfectly calibrated, but looking at train and test data, so on the right-hand side here, we see that, they align relatively well. So the distribution of all the covariates in the model. But if we move to cross-study validation, we see that, that relationship really breaks down. So it indicates that we have very clear distribution shifts in our covariates between train and test data when we separate studies. So that explains why it's so hard to do extrapolation, because we're doing predictions out of distribution. I'm sure many of you have come across this in your models, perhaps on a smaller scale. But also another issue related to the uneven distribution of data that we saw in one of the earlier slides is that, some countries have very little data. So this means that there are quite large discrepancies in the estimated performance between different countries. And because these indicators are going to be used potentially by countries or organizations within countries, that has some implications for users, that we have these big discrepancies. So to summarize these key results, with the risk of stating quite obvious things to this crowd, systematic testing is obviously very essential for any models that are being used in the real world. And data, as you know, is the fundamental constraint on model performance. So we could have used more, sophisticated, fancy models, but I expect the results would have been very similar. And I think we can achieve decent interpolation in the right circumstances when we have enough data, but true extrapolation to entirely new contexts, that's really an open question. But still, model design matters. So explanatory power does not equal predictive power, which is important to remember when designing models. So just to end with some implications or recommendations, I would say in general, any kind of top-down sort of indicator should generally be treated as directional, unless there's really proof that it's good at making predictions, and not viewed as sort of spatially precise. I think it makes sense to try to downscale things to be less top-down where possible, where we can get sort of better data that might not be sort of globally available. But we clearly need more funding to scale standardized biodiversity data collection, as many have pointed out before. And then some priorities in terms of us as a modeling community. I mean, there's definitely more data out there, but it's still scattered for this type of sort of global model. We need to prioritize the contribution of large study data to these repositories like GBIF. And I also think that there's a case for investing more in advancing these top-down macroecological models as a complement to species distribution models. And with that, I want to thank my collaborators and Biodiversity Data Lab, and thank you all for listening.
Thank you, Jakob. We've got about 1 minute for a question. We're running a bit behind, but we'll try and catch up.
Hi, Jakob. I really enjoyed your talk. So there's been some work in geography coming out that says we shouldn't expect replication across sites. Like in geography, we should expect weak replication. So I'm wondering if in your cross-study validation, which is the kind of like geographic replication, like is the gold standard to be able to do that, or should we be looking at context between sites and not really expecting those models to perfectly predict what's happening in another place?
I think that's it's a really good question, and I wouldn't expect good extrapolation performance. I think, to me, getting better overall model performance is really about collecting data such that we are more often in the interpolation case than sort of having to make true extrapolation. I think it might be possible in some circumstances, and maybe there's potential work in terms of model architecture and so on, but generally out of distribution predictions, it's extremely hard when you have learning models.
Cool. Thank you so much. We'll leave it there. Please thank Jakob. So our next presenter couldn't be here in person, unfortunately. They're from Australia also, Simon Ferrier. So we'll be watching them virtually. They'll be talking about a model-based global indicator of the capacity of ecosystems to retain biodiversity under climate change, the Bioclimatic Ecosystem Resilience Index.
Good morning, everybody. My apologies that I was unable to join you in person for this session, but I hope the conference is going well for you. The Bioclimatic Ecosystem Resilience Index, or BERI, is a model-based indicator of the capacity of landscapes to retain species diversity in the face of climate change. And it does this as a function of the area, condition, and connectivity of natural ecosystems. The BERI was originally developed under funding from the Biodiversity Indicator Partnership's Mind the Gap initiative back in 2017. And this was to fill a gap that was identified in the CBD Aichi Target Indicator framework. It's now recognised by the CBD as a component indicator for the Kunming Montreal Global Biodiversity Framework under Target 8. The BERI is generated at 30 arcsecond resolution, a little bit finer than 1 kilometre resolution across the entire land surface of the planet, excluding Antarctica. And it's also being increasingly derived at finer resolutions using best available national data sets. In this presentation, I want to focus particularly on how the BERI can be applied in biodiversity assessment and planning. But first, let me give you a sense of how it is derived by stepping through this simplified workflow. Derivation of the BERI is underpinned by modelling of spatial variation in the present-day species composition of ecological communities and of expected shifts in composition under a plausible set of future climate scenarios. For the global implementation of the BERI, this has involved applying generalised dissimilarity modelling, or GDM, to occurrence data from GBIF for more than 400,000 species of plants, vertebrates, and invertebrates. This modelling is then combined with best available mapping of present-day ecosystem condition derived from remote sensing. For the current global implementation, this involves modelling the probability of occurrence of different land use classes as a function of continuous land cover attributes derived from remote sensing. And to date, we've relied mainly on using vegetation continuous fields data from MODIS. These models are trained using coarser resolution estimates of the proportional coverage of the relevant land use classes from the Land Use Harmonisation Initiative. And the process is therefore a form of statistical downscaling. Predictions of the probability of occurrence of the different land use classes for each 30 arcsecond cell on the planet are then translated into a continuous measure of ecosystem condition using scaling coefficients generated by the Natural History Museum's PREDICTS initiative. The BERI then assesses how well present-day areas of habitat are connected to areas expected to support a similar assemblage of species in the future to that currently supported by the location of interest. Assessment of connectivity across space and time involves combining fundamental principles of metapopulation ecology with least cost path analysis. The details of this approach are beyond the scope of this presentation, but the approach is fully described in the paper at the bottom of the screen, which is, I might add, also the primary reference, the original paper describing the whole methodology of the BERI indicator. The indicator generated by this analytical process can be used to monitor change in the expected capacity of ecosystems to retain species diversity in the face of climate change as a function of observed, that is, past to present, changes in ecosystem condition. We can then map the results for the BERI at raw grid cell resolution across the entire land surface of the planet, but these results can also be summarized and reported by any desired set of spatial units. Currently, we do this largely by country and by biome within each country. This chart is an example for PNG, showing change in the berry from 2000 to 2020, and reporting that by each of the major ecosystem types or biomes within PNG. Even though this chart is showing change just up to 2020, we are about to make available updated results reporting annual change in the indicator for all countries and biomes of the world from 2020 right through to 2024. These results are also being made available through an increasing number of other portals besides CSIRO's own portal. And for example, results are already available at raw grid resolution through the UN Biodiversity Lab, and we are now working with the Bon in a Box initiative to also provide access to results through that front end. The berry can also be employed as a leading indicator, thereby informing planning of area-based actions such as protection or restoration. This involves predicting the change in ecosystem condition expected to result from such actions and the expected flow-on consequences of this change for the capacity of the system to retain species diversity in the face of climate change. Here is just one example from several years back of the application of the analytical approach underlying the berry to prioritizing the location of new protected areas across tropical regions of the world. And that was work funded by the GEF, undertaken in collaboration with Conservation International. As I mentioned at the start of this presentation, the berry is recognised as a component indicator for Target 8 of the Kunming-Montreal Global Biodiversity Framework. But the integrative nature of this indicator means it can potentially play a much more extensive role in linking components both within and across multiple targets and goals of that framework. So, as shown in this diagram, within goal A, the berry can actually help to better link the ecosystem-focused and species-focused components within goal A. And then looking more broadly, the berry offers this potential to also better link actions implemented under targets 1 to 3, that is spatial planning, restoration, and protection, to better link the planning and implementation of those actions to achievement of outcomes under goal A. Widening the focus even further, the berry also offers potential to better link efforts across the CBD's Global Biodiversity Framework and the UNFCCC's Global Goal on Adaptation. The Global Goal on Adaptation was first established under the Paris Agreement, and there has been a big effort over the past 2 years to identify and develop a list of indicators for reporting on progress towards achievement of the global goal on adaptation. And as a result of that process, there are now 100 indicators that have been identified by the UNFCCC for potential adoption at the upcoming climate COP30 in Brazil in a couple of months' time. And berry is one of 10 indicators on that list, one of 10 indicators under the ecosystems and biodiversity theme. And it's worth noting that that process towards coming up with that list of indicators started with more than 9,000 candidate indicators across these various themes, and the process had to reduce that down to a maximum of 100 indicators. But the berry is on that list, which opens up considerable potential for it to be used to better link efforts across those two big initiatives. So far, I have focused mostly on the global implementation of the berry, but as I mentioned at the start, there is also considerable potential for this indicator to be derived and applied at finer spatial resolutions. Within Australia, this finer scale application has been particularly prominent recently in the release of the National Climate Risk Assessment for Australia. And also we, CSIRO, are now working with both South Korea and Canada to help institutions within those countries derive the berry using best available national data sets for South Korea and Canada. This application of the berry at finer spatial resolutions can also be extended to the subnational scale, and just one example here from the state of New South Wales within Australia, where we have worked with the relevant state agency to derive the berry using best available subnational data sets. In conclusion, the berry offers a powerful means by which to assess the expected impact of changes in ecosystem area, condition, and connectivity on capacity to retain species diversity in the face of climate change. Its predictive nature can help link more strongly planning and monitoring of area-based actions, for example, under the GBF's targets 1, 2, and 3, to the achievement of outcomes for biodiversity under the GBF Goal A. The berry is already operational, reporting annual change across the entire land surface of the planet, both by country and by major ecosystem types or biomes. For more information, you can go to either of those links at the bottom of the screen. Thank you very much for your attention and best wishes for the rest of the conference. Thank you.
So Simon has joined virtually online at 3 a.m. in Australia. So if you have questions, please put them in the chat or reach out by his contact details. So next up, we have Andreas, who will be talking about, actually, no, sorry, wrong person. Sorry. We will have Harold Horsley, who will be talking about testing a terrestrial biodiversity model in the ocean, hierarchical Bayesian beta diversity modeling with simulated plankton communities.
Hello. Thank you. Hello. Yes, thanks for inviting me. I'm Harry. I'm at NTNU in Norway, and I'm generally working on biodiversity modeling in the ocean. But this piece of work is about testing this terrestrial diversity model in the ocean. And it's actually the model that we just heard from Simon about. So yeah, that's the question we're trying to answer. Can we apply this model? And on the left, we have a predicted map of the community composition of vascular plants in Tasmania in Australia. And we see similar colors representing similar communities. And it's a very static map. And on the right, we have predicted chlorophyll A at 20-minute time resolution along the coast of Norway. And we can see how dynamic this system is. And the question we're trying to answer is, in such a dynamic system, can this model perform well? And quickly on why we would focus on plankton, although I think I didn't mention that, we're focused on plankton. They are organisms which are very small and they drift in the water column, and they compose 90% of ocean biomass. And they also sequester 40% of global carbon. So they're very important ecosystem components. And the problem is that there isn't much species level data of these of them. You know, we can, when we take genomic data, a lot of the species in that data is new to us. So we've taken a community-based approach because of how sparse that species level data is. And communities are the collection of species or traits in a particular area. And much like in SDMs, if we correlate the environmental factors to these communities, we can predict diversity. And so, and why would we want to do that is so we can decide where to build, where to fish, where to protect, as we all know. So generalized dissimilarity modeling, as Simon just mentioned, it's a type of generalized linear model, and it takes static maps of diversity from gridded predictors. And these predictors are usually time averaged. So in the case of vascular plants here, we have yearly average temperature, yearly precipitation, elevation, and then we can compare communities pairwise and we can predict a map. And what our work has been focused on is taking a time average of a whole year, especially in Norway, where it's very, very seasonal. You get no light in the winter, and phytoplankton depend on that. So we took a snapshot in time to see how well this can perform in just the dynamic situation of one time step. And so these are going to be instantaneous in a time. And so a little bit under the hood on the model itself, say we have this map in space, as we see on the left, where we know some community at 3 locations, I, J, and K. But we know the environment everywhere. It's standard modeling stuff, as we all know. And we're trying to predict this dissimilarity between I and J and I and K. And we also have the observed between at those locations as well. And the modeling really becomes minimizing the difference between these two. I'm sure most people here are aware of that. And then we can predict out of sample with this learned function F, this mapping of our prediction from our environmental variables. Something that I'm not going to talk about too much until the very end is that we actually used a Bayesian hierarchical version of this model so that we can get out uncertainties. But it's not so important for the next bit of the talk. So this approach, this time average environmental variables, we're changing to these instantaneous variables. And so from this point, we ask, okay, what data can we use to test this? Data is very sparse, the ocean is very big. And what we've done is we've taken an ocean simulator. So this ocean simulator takes in wind, weather, light, and fresh water, and it outputs these physical and biological processes. It calculates them with partial differential equations. And so you can get out, as we have here, chlorophyll A, but you can also get temperature, salinity, current speeds, nutrients, and even plankton functional types. And so the resolution we've used is 800 meters by 800 meters at a daily resolution. So that's going to get us our environmental variables. And here we've used temperature, salinity, vorticity, which is a measure of rotation, which is important for eddies because these capture a lot of diversity. And then also nutrients because they impact the growth of plankton. And then we ask, okay, what plankton data to use? And we mentioned yesterday about how important simulation data is. And we're actually also taking simulation data from the model. And the reason we might want to do this, particularly when we're trying to test, it's good because we can generate arbitrary amounts of data. We can get the true accuracy of the model, and we can also assess our data requirements quite cheaply. as long as the model is, a decent approximation. We're not claiming that it can perfectly represent diversity, but we are seeing if this model can represent dynamic ocean processes. And so that's what we did. Here we trained on 100 sites, so just under 5,000 pairs at one time index. So in the middle of July in this case. And we learned our function f, and then we produced this map here. The similar colors represent similar communities. And the dream of what we want to do is, once we have learned this, we can predict through time because we have our ocean model. And that's what we're seeing here, is each time steps 1 day, and we're seeing the community shifting as the ocean is moving. And that's along the coast of Norway there. And so our question is, okay, so we've done this, cool. Is it accurate? And how well does it go across time as well? And so that first question, how accurate are the predictions and how well does it generalize in time? So this first one, results. So here we're looking at model skill along the y-axis. And then on the x-axis is number of training sites. And model skill is a relative measure of how well your model predicts versus some null model. And in this case, the null model is just the mean of your training sites. So higher is better. And we see, in the words of Jakob before, we see the flattening. And as in the words of Jakob, that's because I've forgotten the words of Jakob. The fixed effects, that's it. So the fixed effects have kind of learned everything we can from them. And in this case, we see the R-squared values in the legend. About 0.5 for the R-squared value, 0.6 for the summer. And that's quite a high result in ecological systems. We might expect that given that it's a simulation and we have all the variables. But it's quite a strong result. And the last point is just the difference between the summer and the winter. So the summer is the orange line and the winter is blue. And blue is lower because in the winter there's very little activity. So the null model is actually quite a good predictor. There's just not much diversity. There's just not much dissimilarity. So that's one time step, tested on 100, trained on various and tested on 100 sites, on 100 held out sites, because we can do that with our simulation. And then moving on to how well does it generalize in time? Well, here we see we trained a one-time step and then we tested weekly. And now we've shifted to error on the y-axis. So the lower is better. And we see that date, 15th of July, where we have quite low error, but it rises very quickly outside that training window, as you see different conditions and different communities throughout the year. And error improves again in the winter, but again, this is due to the low diversity in that period, we presume, because it's quite easy to predict there. Moving to now the 15th of February, a winter training site, test on the rest. We see it really exploding in the summer again. But it's in the similar conditions of the winter, it's fairly decent at prediction. So to conclude on that, we can say, look, it's fairly solid performance when we predict in one time step, as the model is designed to do. But when we're dealing with time, as we are in the ocean, and particularly plankton, which are completely driven by ocean currents, they're completely driven by time. These trees on land have timescales of decades, plankton have days to weeks. So you really need to capture time in some way. So from this, we'd like to, work on real data using satellites and genomic data, but also see what we can do, how we can change the model. So incorporating time as a predictor, temporal difference could be interesting, and predicting across multiple time steps, maybe that can sort out the time generalization. And one thing I'm quite excited about, and if anyone has any background in this, is the water parcel history or Lagrangian methods. So instead of just this instantaneous temperature salinity on your predictors, over the last days or weeks, where has that water parcel been? What has been the growth conditions for the plankton? And you can kind of track that with satellites and simulating particles. And so that's some exciting work that maybe I will get into. But also what I'm really excited about is optimal sampling. So as I said before, this is a Bayesian model, and it produces uncertainty, and we'd like to leverage that uncertainty to produce optimal sampling sites. Because really, we want to close the loop, I think. This is not a model sitting on its own. It's really part of a greater system. So we have some data collection platform on the left, and because the model is data-driven, we improve the predictability of the model. And then we generate some map. And that map is wrong, and it will always be wrong. but we would like to improve it. And so with uncertainty, we can say we're uncertain under these conditions, at these times, at these locations, and 1 or 2% efficiency gain in sampling sites, or maybe more, is going to be a huge difference over decades. And so this doesn't just restrict to the type of model I've used. As long as it is, has these 3 properties, it is data-driven, it's generative, and it's, provides uncertainty, you can do this system. These are some references. I'd like to point to the final one at the end, which is the model I would use, the spatial generalized dissimilarity mixed model. And thank you very much, and very happy to take questions and talk to anyone after who's interested. Thank you.
Thank you, Harold. We do have time for a question. Thanks, Justin.
Yeah, thank you. Just wondering, so here you've used the simulation model to produce output that you can test your statistical modeling approach on.
Yeah.
Do you see a role eventually for the process-based simulation in what you're really trying to get at, or would you just want to go towards the data and the statistical model?
Yeah, that's a big, they're kind of complementary approaches, I think. And there are, there's a lot of these ocean models. MIT does one, and they have a lot more phytoplankton types. So there's definitely space for that. And particularly if you can improve the prediction of the currents, which there are some deep learning approaches that are coming that are for weather that will move to the ocean, which will be really amazing. So definitely there is space for that. I would like to use this for genomic data, so eDNA metabarcoding, because that's such a huge picture of the diversity you have. And maybe you are taking these predictors at those locations, and then you can use the ocean model to then predict in space with that genomic data. Because this, phytoplankton types, genomic data, you're getting hundreds of species. So that would be cool. But yeah, both approaches, I think, are complementary. Thank you.
We are running a bit behind, so we might leave it there, but please follow up with questions, and please join me in thanking Harold for a wonderful talk. So next up, we have Andreas Schwarz Meyer, who will be talking about improved forecasts of biodiversity exposure to climate variability and change using decadal climate prediction.
Hi, everyone. I'm Andreas Schwarz Meyer. I'm a researcher at the University of Cape Town, South Africa. I'm here to talk a little bit about the work we've been doing using decadal predictions to forecast biodiversity exposure to climate change. So we all know that climate change is affecting biodiversity at all levels, and what we've been observing at these impacts are arriving earlier than we were expecting and at a higher intensity. So for decades, the ecological community has been trying to build ecological forecasts to predict where and when these impacts might emerge. And one problem is that most of these forecasts so far are built focusing on a few snapshots of the future, usually towards mid and end century. And therefore, they're not really able to capture more imminent climate risk to biodiversity. Recent initiatives try to use a more finer temporal scale climate data, so we can see these changes more like a movie instead of a snapshot of the future. But still, another limitation that we have here is that most of these forecasts are built based on scenarios, usually the IPCC scenario. So they are scenario dependent. And even though these ecological forecasts are very important for us to have a good idea of what exactly might happen depending on what scenario we follow, and to set long-term international policy goals, they don't align well to shorter-term plans or action needed to protect biodiversity from more imminent climate risk. And many conservation practitioners, they have their strategic planning that goes from 1 to 5 years. And if you go to chat with them and say hey, I have an ecological forecast that says that in 2070, those species in your reserve might be exposed to climate change, there's not much they can do with this information at this moment. So to fill this gap, we try to use ecological forecasts climate forecasts that might align better with these near-term needs for biodiversity conservation. And these are decadal climate predictions. So the decadal climate predictions are pretty much the same climate models that are used in the IPCC scenarios. The main difference is that they are initialized with conditions that match very closely the climate conditions we observe. So they're kind of like, in a way, a hybrid between a climate projection and a weather forecast, when they start in the true climate and then they're run for up to 10 years into the future. And because they're initialized based on the true climate, on the observed climate conditions, they have a very good predictive skill, especially for the first forecast years. So here in this example, you can see the black line is the observed global temperature average. And you can see that every one of these little dots are a decadal prediction initialized in a different year. So the first initialized in 55 and then run 56, 57, 58, 10 years into the future. The second initialized in 56 and then run 57, 58, and so on. And you can see that especially for the first forecast years, the prediction that you get of the global warming, of the global temperatures from the decadal predictions is much closer to the black line, the observations, than if you use an uninitialized model, which is this gray line over there. So if decadal predictions offer us a better ability to predict the climate, maybe they can offer us also a better ability to predict risk to biodiversity. So that's what we wanted to test here. So our goal in this project was to assess whether decadal predictions can improve forecast of species exposure to temperature beyond their historical thermal limits. So this is the method we use. We use monthly mean temperature, which was the variable like more easily available for the decadal predictions at a 1 degree resolution, which is the highest resolution they have, unfortunately. We use ERA 5 as the reanalysis climate data set. So ERA 5 was our observed climate data. So that was our reference model. And then we used 2 simulation climate data. One was the decadal predictions here, and you can see that we use 5 climate models with different model runs in each of these models, and we use this for 5 forecast years. So we have like 50 model runs times 5, so we have 250 simulations here. And we also use the CMIP6 historical run as a comparison, because our goal was to test, like, are these initialized predictions better in predicting exposure than the uninitialized prediction? And the uninitialized were the CMIP6 historical run. So we use the same models and the same number of simulations. And then use a time frame between 65 and 90 as our baseline period to estimate our historical thermal limits. And then we use 1991 to 2014 as our forecast period. And we stop at 2014 because it's when the historical run from the CMIP6 stops. And then from 2015 onwards, we have the scenario simulations. And then we tested this for over 7,000 species of amphibians. And to estimate the historical thermal limits, here is the same model that Pep explained yesterday. So for each species, we extracted the temperature data across the entire range for the baseline period. And then we calculated for each month the 99th percentile of the temperature. So basically here we had 12 thermal limits for species, 1 for each month. We're not just interested in the hottest month ever, but also interested in this sub-annual exposure where I can say like, oh, it was the hottest January the species ever experienced, or the hottest February and onwards. And then we estimated to estimate exposure. We just look into our forecast period to 91 to 2014 and say, okay, where and when these limits are going to be exceeded on a month-specific basis. And then from that, we derived an exposure indicator, which was like a very simple way here to summarize all these like months of exposure that we have. And this indicator was simply asking, are these models, the decadal predictions and the historical, able to detect whether or not a species was exposed in a given year for at least 1 month? So could be 1, could be 2, could be 3, doesn't matter. This was our indicator, exposed or not for at least one month. And then to assess the ability of the decadal predictions and of the historical simulations to predict these, we used 2 approaches. One was a confusion matrix of all data set. So these basically for every model run, for every species in every grid cell, in every month, in every year, in every month, we have either a 0, not exposure, or 1 exposure. And then I use all these 0s and 1s and compare with the ERA 5 to build this confusion matrix. That's not usually how the climate people assess the scale of the decadal prediction. This was just my stubborn ecologist brain saying like, I want to do a confusion matrix out of it. And then Breyer's skill score, it's how usually these events are evaluated in climate science because they provide a probabilistic assessment. So here, because we are using many model runs and many different models, instead of just comparing the 0 and 1s from an individual model run, we calculate a probability of an event happening. And this probability is, if let's say we have 10 model runs, and 7 out of these 10 model runs say that we're gonna have at least 1 month of exposure in a year. So we have a probability of 0.7 that this event is happening. And then we calculate a probability for all models, and then we calculate a multi-model mean based on these probabilities. And then once we have this multi-model mean, we calculate the mean squared error between these multi-model probabilities and the observation that we get from error 5. And then we have this, what we call this Breyer score. And then after that, we calculate what is called a Breyer skill score, which is basically evaluating the mean squared error of the decadal predictions and of the historical simulation. That's 1 minus the Breyer score for the decadal predictions divided by the Breyer score of the historical simulation. And if these values are positive, this means that the score for the decadal prediction is higher, so they have higher predictive capacity, and is negative shows that the historical simulation have a higher predictive capacity. So these are our results. This is the median number of years where we have our indicator like across the world. So here's showing that what was the median number of years in which we have at least 1 month of exposure. And this is the median across all species in the grid cell. So you can see that we have lots in South America, in Africa, in Southeast Asia, in Mexico, or like kind of the exposure hotspots for amphibians between 1991 and 2014. These are the results of our confusion matrices. So here in red, we have the decadal predictions, and in green, we have the historical data. And we have 3 metrics here: specificity, precision, and sensitivity. So, and each of the violin plots, of the red violin plots, are showing one forecast here. So you can see that overall here, see that decadal predictions are better in identifying the non-exposed species and in avoiding false positives. And the historical simulations are better at capturing true exposure, but this happens because they tend to overestimate exposure. And this is the comparison of the Bryan skill score now with the multimodal mean. So here I calculate a Bryan skill score for every species in every grid cell in every year. So these yield like roughly 6 million Breyer scores. And from all these Breyer scores, roughly 80% here, the blue color, showed that decadal predictions had a better skill in predicting the exposure than the historical simulations. In roughly 18% of the cases, the historical simulation had a better performance, and in roughly 2%, the skill was very similar, so the difference was less than 0.1. So here's showing that decadal predictions do have like a larger predictive capacity. And this plot shows, so for every species in every grid cell in every year, I have a probability that exposure occurred in a year. And what I did here was that I grouped these probabilities by year, and I summed them just to show how they change along the year. So you can see in the first plot that at forecast year 1, the gray line is the observed exposure from era 5, and the red line is the cumulative probabilities from the decadal predictions. You can see that it follows very closely the observation when we sum the exposures that happened every year. Forecast year 2, we still have like a good alignment, and from forecast year 3 to 5, we had a decrease. But still like a little bit there, 4 and 5, like not that much. But then in the bottom, we can see the same for the historical simulations. So you can see that the historical, they're very good at getting the trend. They do get a trend right, but this interannual variability, unfortunately, can't capture. This is a map showing where the skill is higher for the decadal predictions and where the skill are higher for the historical simulations. So you can see that in the vast majority of the communities we analyze, or the grid cells, on average, the skill of the decadal prediction was better, especially in the places where exposure did not occur. Remember that I said they are very good at identifying non-exposure events. So here, side by side, the map with the number of the exposure with the indicator that we derived, the median number of years where exposure occurred, and the other one is the decadal prediction skill. So you can see that in places where we have exposure is that we start to observe a decline in the skill from the decadal prediction, but that's natural. As more and more exposure happen, it gets more tricky to capture this correctly, but we still have a higher skill compared with the historical simulations. But in a few places like in Paraguay, Argentina, in Mexico, in Northeast Africa, and in Southeast Asia, we still have some places where the historical simulations perform a bit better. So our key findings is that decadal predictions do improve the skill of biodiversity exposure forecasts compared to the CMIP6 simulations. They perform better because they are good at identifying non-exposed species and avoiding false positives. However, they might still miss some true exposure events. But if we're thinking about that these type of predictions could be used to prioritize resource allocation and monitoring, actually what we want to do is we want to avoid false positives, to not allocate resources in areas that might not experience exposure. So decadal predictions do provide a promising basis for biodiversity forecasts at actionable timescales. And this is a work in progress. So in development, we have right now assessing the skill of additional exposure indicators. And that's one of the feedback we would like from people, just what type of indicator do you think would be useful for the organism you study? And then we can assess the skill of these indicators. We are developing scientific software to scale up these analyses and be able to have these predictions for hundreds of thousands of species. And we also are developing a pipeline to automate these analyses, because these decadal predictions are produced every year. So we can automate these analyses and every year generate predictions based on them of biodiversity exposure to climate change. Thank you.
Thank you, Andreas. We got time for a question. Any question? I have a quick one. What sort of other exposure indicators are you exploring?
Yeah, so one is the actual number of months of exposure we have in a year, and not just whether or not it was 1 month. The other one is the percentage of the species range exposed. And those are and the others, that's kind of we call a time integrated exposure, where across the range for all possible months of exposure that you can have, what's this area under the curve, or what's the percentage of them you have. So these are the ones we work at the moment.
Very cool. I'll look out for it.
Cool.
Thank you. Please thank Andreas. Okay, so now we are moving into the section of the program where we have some 5-minute presentations. So first up, we have Santiago, who will be talking about empowering abundance-based species distribution models with the new ADM package.
Hi, everyone. Buenos dias. So this is a very quick talk about the new ADM R package. So was explaining what SDM are, so I will not explain what SDM are, but SDM could be constructed based on abundance data, or one expression of abundance on density of individuals. So this imposes some difficulties at the moment to construct models, because abundance on density could be expressed between 0 and 1 or 0 and 100, or unbounded, positively unbounded, between 0 and infinitum. So this is the reason that we created that package, one of the reasons. ADM package follow the same FlaxSDM package. There was a previous package that we created a couple of years ago. So the main objective of this package is provides modelers with different building blocks, our functions. So you construct your modeling workflow combining in the way that you want these individual functions. So it is make very, very flexible and integrated with other package, the modeling protocol. So in the same way, ADM can be easily integrated with FlaxSDM and maybe with other R package. For example, to calculate model exposure and model extrapolation, delimit calibration area, or do some data partitioning based on abundance data. Up to now, this package have 35 functions. They are grouped in 3 groups: modeling, that we provide a bunch of functions to fit and validate different kind of algorithms. Then we have the post-modeling, that basically a function to predict this abundance-based bet, and other fancy interesting partial dependent plot and bivariate partial dependent plots. And then we have miscellaneous tools that is a bunch of functions to help modelers to construct this modeling protocol. One of the main features, up to now, we implemented 9 algorithms, from correlative GLM to machine learning like DNN and CNN, convolutional deep neural network. Okay, because you are modeling with abundance, is your response variable, so you maybe sometimes before some algorithms is better if you transform or make some transformation. And then one of the things that I highlight more is the simplicity of the outputs. So we don't create any weird and ugly new objects. So all the outputs of the function always will be a raster, a spatial vector, a table that is a fancy data frames, or a list of combination of the previous object. So this make it easily integrated with other package, and you can easily explore all the output of each of the functions. In modeling functions, we provide these 2 family functions, fit abund and tunabund. Fit abund just fit and validate models based on default hyperparameters, and tunabund you can perform fine tuning based on brute force approach. This is the list of algorithms implemented. And for convolutional network and deep neural network, we created a really interesting functions to create networks. So it helps modelers to tune these time-consuming algorithms. This is the list of metrics, sorry. Just some examples of partial dependent plots based on univariate or bivariate partial dependent plots. So in these regions, you can see the extrapolation. This is the calibration condition, and this is the calibration condition, extrapolation condition. So you can learn a lot with these type of plots and how weird they are. That's all.
Thank you, Santiago. A very quick question while the next presenter comes up. Yeah.
Just a quick question on the deep neural networks in your package. Are they implemented natively in R, or do they like call wrappers that call Python underneath, such as TensorFlow or PyTorch?
PyTorch.
Okay, yeah. Great. Thank you.
Thank you. Please thank Santi. All right. So next we have Lucas da Cunha Godoy. Sorry, pronounced that terribly. Talking about DRM, a Bayesian approach to dynamic range models in R.
All right. Hello. Okay, it's working. So you have 10 seconds to scan this QR code. My name is Lucas. I'm a postdoc in the Global Change Research Group at UC Santa Cruz. Today, I'll be talking about this package that I've been working with in the postdoc, and it's called drmr. It's a package to fit dynamic range models in a Bayesian framework. So the dynamic range models, they are a class of models that combine statistics and biological realism. So I'll talk a little bit about it today. So I'm mainly advertising the package here and hoping people will use it. I'll not show any functions. So as we have seen today, extrapolating is hard, but it's still of extreme importance in ecology, for management, for conservation, and many other things that you know more than I do. The usual tool to achieve that is the SDMs. And as with any other model, again, extrapolation is really hard and the SDMs have been reported to struggle under future novel conditions, such as the ones that we experience these days in our little planet. And they also lack mechanism which can help to mitigate or alleviate that struggle. There's another implicit assumption there that the species that we are studying are in equilibrium with the environment that is often violated. So there's been a push in the literature to kind of improve these forecasts to be more realistic. And in this little hexagon here, we have environment, physiology, demography, dispersal, species interactions, and evolution. So most of the time, the SDMs only take into account the environment. And we believe that we can do better if we take other things into account, and that's what the package does through an age-structured model. You don't need to have age-structured data. It's just like the model has assumptions, and you get to choose how many age groups your species has, how many of those age groups actually move in between patches, and which age groups you actually observe. So these are some features of the package. It also allows us to estimate relationship between environment and some interesting demographic quantities, such as recruitment or mortality rates. So I have 2 case studies in a paper that is currently under review. One of them is with summer flounder, not really a charismatic species, I would say. And the other one is the red-bellied woodpecker, but they are probably best known for this other pictures here. Although that's not a red-bellied woodpecker, but who cares? It's cool. So what we are looking at, all right, is some model fit and forecast from the DRM or package using drmr and an SDM, basically zero inflated SDM that is usually used in practice, which is on your right-hand side. On the left-hand side, we have a DRM fit with the environment influencing recruitment. In the middle, we have a DRM fit with the environment influencing mortality rates. On the x-axis here, we have the time. On the y-axis, we have density. And each row in this table in this plot is a site from the study chosen at random. The horizontal lines represent the point where we start doing out-of-sample predictions instead of just fitting. So we can see that the DRMs seem to capture better the nuances in the data due to its structure, I believe. Just looking qualitatively, I can say that, but quantitatively, if we look at prediction skill, it's much better, at least for this case study. I don't expect it to be always better, but I expect it to be a useful tool for modelers. So it also gives us some other interesting information. Here we are estimating the relationship between sea surface temperature and recruitment for the summer flounder on the left, and sea bottom temperature with survival on the right. So we can basically tell in which sort of temperatures these species thrive or do the opposite. So to wrap it up, I hope this package will lower the barrier for ecologists to access this class of models. It is user-friendly, but well, this is my opinion and I wrote it, so let's see. And in addition to generate more robust forecasts, it will let ecologists to test which mechanistic hypothesis may be actually driving the effect of climate change on species. So the package is open source and will be available on GitHub once the review process is over, but that QR code in the beginning gives you access to all the code to reproduce the results and check it out. All right, thank you.
Thank you very much, Lucas. We don't have time for questions, but please find Lucas after. Our next speaker is Jamie Kass, who will be talking about getting a handle on model complexity and extrapolation when making future forecasts with biodiversity models. Thanks, Jamie.
Hi, well, this room is very full. My name is Jamie Kass. I'm an associate professor at the Graduate School of Life Sciences at Tohoku University in Japan. And my talk is called Getting a Handle on Model Complexity and Extrapolation when Making Future Forecasts with Biodiversity Models. I'd like to thank the organizers of these 2 sessions for organizing everyone together and getting to talk about great new cutting-edge methods for biodiversity modeling. So I probably do not need to describe ENMs to this crowd, but here goes. So ecological niche models, as they're called, make predictions of niche relationships between species in the environment, and also make predictions of potential distributions for species. They use records of species occurrences and also pseudo-absence or background data, or absence data if you have it, and also environmental predictor variables. And a very nice thing about them is that you can make new predictions for environmental data for times or places where there are no samples. So typical application would be making predictions of range shifts for climate change. And then you can also make predictions like this, which are marginal response curves, which show the species' response to the environment. And so I'll be using these to demonstrate some examples for improving our interpretations of model extrapolations. So what is the aim of this response curve? So these response curves ideally should estimate realistic niche relationships. And this means that there is a peak to the curve, and then it descends, and eventually the curves go down to zero, or the tails go down to zero. And so the gold standard for this, as Natalie explained yesterday, uses mechanistic ecological niche models, which employ physiological data, and then you specify the response a priori. However, these require experiments and mathematical models. And so the problem is we don't have this for most species. Most species are data poor. And so we end up using correlative models instead. So when we're using correlative models, we should still aim to model species responses that resemble these physiological limit curves, especially when we're doing model transfer, which is making predictions into unknown predictor space. And so machine learning methods can be optimized by tuning them to, machine learning methods can be tuned to optimize model complexity. You can run many different models and test them and see what the optimal complexity is. However, they can also lead to unrealistic responses that make constrained extrapolations. And so what are some strategies for improving model responses? One of them is to map environmental similarity between the training and testing conditions over space. There are multiple methods to do this. You can do this to visualize areas of extrapolation, and you can also do this to limit predictions to interpolated areas. Another strategy is to use kind of a new method in ecology, which is explainable AI. There are tools to make simple models for point predictions in space to estimate complex model behavior. So basically, you take a point in space, and you make a simple model that estimates the complex model behavior, and then you can get these little plots of variable importance. And so when complex models are used, which they often are, it can help identify regions behind unexpected predictions. They're relatively new to ecology, but they'll be more important as we make as we use more and more complex models to make predictions. And there are many opportunities for investigating extrapolation. Strategy 3, sometimes when we have unrealistic response curves, we might want to fix the response curve. And so in this example, you have sparse sampling over space, and we made more thorough background samples for high mountain areas, and we're able to fix the truncation in the response curve. So that's good. Thorough sampling can remedy things, but environmental weighting might also you can also use environmental weighting to balance undersampled values to get more extreme environmental values with higher weights. Another more extreme example is to use environmental background sampling. This includes extreme values outside the training data to force the curves to fall down to zero. Another way to do this is to use custom clamping. So clamping is to limit the new predictions to extremes of the training data. So even if we have realistic response curves, some algorithms can only clamp. So machine learning algorithms only clamp. So custom clamping can align these predictions with our expectations. And these decisions are based on the response trajectory. But the effect depends on the density of the transfer conditions. And lastly, I have basically no time, but there are great potentials for deep learning. And you can probably read the rest of this slide. But basically, deep learning algorithms can make very good predictions when you make them very big, when you increase the complexity. And so there are great potentials for making good extrapolations with deep learning algorithms. Thank you very much for listening, and I probably have no time for questions.
you. Thanks, Jamie. Please find Jamie at the end of the session for questions. There was a lot of exciting things in there. Right, next up we have Catherine Villeneuve, who will be talking about BATIS, a benchmark and framework for uncertainty-aware species distribution modeling. Thanks, Catherine.
Oops. Okay. All right. Hi everyone. I'm Catherine, second year PhD student at David Rodnick's lab, associated with Mila and McGill University in Montreal. And I'm here to present our recent preprint, which is called BATIS, a benchmark and framework for uncertainty-aware species distribution modeling. So the origin of the name is Bayesian approaches for targeted improvements of species distribution models. But BATIS is also the name of a cute little bird who can be found in the south of Africa. And our use case for this method was that we knew from previous work that training an SDM on a region with noisy and very limited data is hard, and that you can only usually get high quality data on only a few selected locations. So like we see here from plots made from data available on eBird, we can easily see that data tends to be highly clustered around roads or highly densified areas. And we also knew from previous results that including data areas where we had very limited and noisy information directly into the SDMs could lead to a decrease in performance for highly sampled areas. So we had to think about potentially novel ways to integrate these noisy information without degrading performance in other areas. So our first hypothesis for this work was that if we train an SDM solely on systematic surveys and high quality data, we would be able to get an initial model that can capture some like coarser ecological patterns. And then if you were to combine this prior model with the limited noisy information we have in data scarce region through a Bayesian model, then we could maybe be able to improve performance compared to just including this noisy information into the training set of our model. So basically, our pipeline for this model was that for each subsample location, we generated a prior encounter rate from a deep learning model, which also produces an estimate of uncertainty through uncertainty-aware deep learning approaches. And then we combine through a Bayesian pipeline the limited observation, which then leads to a posterior predictive that we can use afterwards to estimate the encounter rate. And this is just a simplified summary of our approach. So we have the prior SDM prediction from a deep learning model. Then we have another deep learning approach that can predict uncertainty, estimate uncertainty. And then we gather the additional observation from data scarce location, and we combine all of this into our BATIS approach. Oh, thank you. So we created a data set which combined data from eBird from the countries of USA, Kenya, and South Africa. And our results suggested that gathering as few as, let's say, if you have a location with as few as 10 observations, we can quickly improve performance compared to if we just included those observations in training rather than using them into a Bayesian pipeline with the prior deep learning model. So in conclusion, our approach can combine broader ecological patterns from a coarser SDM to fine-grained insights. It can improve predictions from regions with limited data and leverage uncertainty to highlight the relevance of local environmental factors compared to coarser environmental patterns learned from a prior SDM. And further works include extending this to guiding data sampling in data scarce location, and also extend this work to presence-only data set because we only evaluated on presence absence. So thank you.
Thank you. We don't have time for questions, but please follow up at the end of the session. The next speaker is Amy Frazier, who will be talking about scaling sustainable land use planning from global to national levels for biodiversity, energy and agriculture under climate change.
Awesome. Thank you, everyone. This work was done in collaboration with folks from Conservation International, Arizona State University, Ohio State University, and the Humboldt Institute. Thank you. Well, I guess I need this, huh? There we go. Okay, so many of you may be familiar with this picture, or at the very least, the targets, the 30 by 30 targets. A lot of the talks in this conference have been focused on how to support our way to 30% conservation. And we've been focusing mainly on protecting biodiversity, right? But alongside that framework are other frameworks happening globally. This is Paris, France, where we're looking at climate change. We have similar conventions around desertification and so on and so forth. So we need to not just think about conserving land for species and biodiversity, but also how do we meet all of these other targets and plan for all of these other land uses, like development, like agriculture, like energy, and how do we prepare for what the future may hold? So planning to make sure that in the future we have land to support our growing population as well as our endangered species under climate change. So we have been working on some global models to address this problem. So to figure out this puzzle of where do we put conservation land, but also where do we plan for future agriculture and energy. And the big takeaway from our global models has been that we actually don't have enough land when we try to do this exercise globally. So we can't fit it all in. That's a problem. Why does that happen? Is it because we're going to run out of land? Maybe, but probably it's more a function of the data sets that we're using at the global scale, the resolution of those. And so our global scale models are really good at identifying hotspots of where we might focus certain land uses in the future, but they're not great for national or subnational decision making. So that is where our objectives come in here. So we're in our project, we're attempting to downscale our global models to national scales that can be used for decision making and planning. We're integrating lots of diverse living data, but also data on agriculture and energy projections with climate change scenarios to understand how we need to think about these things as our world is changing. And then importantly, to identify conflicts. So when we're thinking not just about conservation, but also agriculture and energy, where do we have conflict between those that we should really be putting our efforts now and into the future? We are doing this work here in Colombia. We have a mega diverse country here. We have lots of pressures on land uses, but also lots of great data to do this type of modeling with. Okay, so in this figure here, it's not a species distribution modeling workflow. Instead, it's a prioritization workflow, which many of you may be familiar with. The conservation targets are what go into a lot of these models that we are familiar with. We're getting our biodiversity data from Biomodelos, and we're also looking at carbon targets. But the bottom there, the development targets are where we're bringing in data on the agriculture and energy. We're minimizing costs while trying to meet those objectives. And then we're planning under multiple different kind of types of scenarios, planning scenarios. So what if we try to plan for nature first? How does that change when we prioritize development and try and kind of fit nature in with what's left? Okay, so some results, global versus national. Everywhere in red is a place where the global model disagreed with the national model. So you can see that's a lot. So our national scale models are much, much different than what we're getting at the global scale. There's more conservation land, that's why that one is lighting up a bit more, but we have red on both of those maps. This is a comparison. Now we're just looking at the national scale. This is a comparison between that scenario where we prioritize nature versus the scenario where we prioritize development. You can see that they're pretty similar, but there are some hotspots on that map where we see those differences. Importantly, when we prioritize nature first, we see about, I think, 50,000 square kilometers more land dedicated to nature, but the amount that we need for development, agriculture and energy stays about the same. So we can actually get, we don't lose anything by prioritizing nature first. In fact, we can fit it all in even when we do that. Okay, lastly, those conflicts. So where do we expect conflicts to happen in the future between conservation, agriculture, and energy? The important color to look at here is the black. The black is where we have land that we need to reach 30% and protect species, particularly endangered species. And that's also land that's good for agriculture or good for energy, like wind energy or hydro power. Excuse me. So those black areas are places that we can put a little more concerted effort to work out what might be the best land use in those places. Excuse me. And with that, I'm out of time, I think. So thank you. This is our entire team, and I don't have time for questions.
Thank you, Amy. So our final presentation in this session is actually a virtual one. We have Mike Carney talking about theory and ecological trait data standards that can improve the utility of functional trait databases. It is actually a 15-minute talk. There was an error in the program. But I might ask if after this last talk, if the speakers could all hang around, because I know there were heaps of questions that we didn't get time for. So that would be really nice.
Greetings from Melbourne, Australia last week. I hope everyone's having a great time at the meeting. Sorry I'm not able to be there, but it's great to have this opportunity to give this talk about how we can take theoretical developments in how we model organisms together with technical developments in how we organise data to improve the utility of functional trait databases. And I want to acknowledge my co-authors, who you can see listed below, the second one being one of the organisers of this symposium. And this is a work in progress, but we're getting close to finishing it. So it's exciting at a meeting like this to think about all the different kinds of data we can now link together about biodiversity, all this living data, through persistent identifiers. And in this web of data, one of the most challenging ones, I think, is the trait data. Now, the trait concept is really anything you can observe and measure about an organism. So it's so broad as to almost be as useful as the concept of a thing. But there's been a lot of thought conceptually and technically about what we do with trait data, how do we deal with it? And that includes, what do we include in the first place? For instance, do we include habitat aspects of an organism in a trait database or not? What level should we organise the data, and how do we deal with these different levels of organisation, from individuals to populations to species? How do we prioritise trait data? How do we know what trait data we need? Where are the gaps? How do we standardise it, including what metadata do we need, and how do we put it all together and connect and store it? So, as I mentioned, there's been conceptual and technical work on this problem. From a conceptual point of view, a really important step was this paper by Cyril Viol and colleagues in 2007, Let the Concept of Trait Be Functional, which is really emphasising that we want traits to link to performance of organisms, to growth, reproduction, survival. And more recently, my colleagues and I wrote a paper that is kind of following on from the latter paper, called Where Did Functional Traits Come From? The Role of Theory and Models. And we were looking at this from the perspective of modelling organisms as dynamical systems, using something called dynamical systems modelling, which wasn't familiar to me through my undergraduate and PhD training, but it's a kind of model that's used a lot in physics. And what I'm going to do now is give you a sense of what these kinds of models are and how they tell us what functional traits are, because they demand parameters, and those parameters link to functional traits. So a dynamical systems model, I'm just going to check on recording, yes. A dynamical systems model involves state variables, and those state variables evolve through time. So we have a rate of change equation of the state, and examples of state variables might be the body size or the body temperature or the water content. These are not traits, but states. And so these traits evolve through time, and we can crank this model and work out how those things are changing, and from those get things like growth, reproduction, and survival, making them functional in the sense of the Viol paper. But of course, we also need environmental forcings, so things like food, temperature, and humidity that drive the model through time and make things change. So there's various kinds of things we can model with dynamical systems models. We can model behavior, we can model heat budgets, but the most fundamental thing we can model is the metabolism of the organism. You can think of it as the energy budget, the dynamic energy budget. And there's a theory called dynamic energy budget theory, which is a really powerful way of modeling the organism as a dynamical system from first principles of physics and chemistry, where it treats the organism as a set of macrochemicals that are evolving through time, food being converted into biomass, and the metabolic waste products and heat being produced. And what we get out of this kind of model is the mass of the animal through time. But you can see this is the mass broken up into different state variables. In particular, we have in green the structure, in grey the reserve, in brown the stomach contents, and in pink the reproduction buffer, or the eggs, egg material growing in the organism. So that's what we get out of a dynamical systems model of the metabolism, out of this DEB theory. And to give you a little bit more of an understanding of how it works, and this is going to be superficial because there's not much time, but the first process is food being converted to the reserve. So that's a chemical transformation called assimilation, feces being produced as a result. And this is, you can think of reserve as a metabolic substrate, which is used to do the rest of the metabolism, including things immediately and also things in the future. So it's not reserve for the only things in the future. It's used immediately and in the future. And so there's a constant flow out of that reserve that is going to, a fraction of which is going to growing the organism, when that's the structure. So the structure is what is built through growth, the process of growth. And the structure also needs maintenance. So there's some of the energy that is coming down this path gets used to pay the maintenance, and whatever is left over is then building new structure. And similarly, not similarly, but the other fraction of the reserve flowing out is going initially to the process of maturation. So the developmental work to organize the organism is explicit in this model. And then once maturity has been reached, the energy is going into the reproduction buffer, so producing biomass in terms of offspring, although there's always a maintenance cost to keeping the final level of maturity that is reached. So that is, in a nutshell, the DEB theory as a dynamical systems model. And it goes through the whole life cycle. So it starts off as an embryo, and the only difference between an embryo and a hatchling in the DEB theory is this part here, the feeding process, doesn't happen, and it's just starting off as pure reserve. So that's a good way to think about reserve. It's what an egg is made of. It's got everything you need to build, in this case, a baby lizard. So that's an example of a dynamical systems model. And of course, all these models need parameters, and that's what this lambda here is. And it's the parameters that tell us what trait data we need to measure. And in the DEB model, or dynamic energy budget model, these are the parameters. There's essentially one parameter per process, but it has a lot of processes that are being considered in a network. So there's a few parameters. And this is where all those parameters link into the system diagram. But the things that we can use to estimate the DEB parameters, these are the basic parameters we must have on the left. There's some that you don't necessarily need, but these are the ones you definitely need. And this is the minimum set of observations you need to estimate those parameters. And they're things like age at birth, age at maturity, length at birth, wet mass at puberty, reproduction rate, things that are quite commonly measured as trait data. And there's a one-to-many relationship between the parameters and the traits. Because essentially anything that you can churn out from the model as an observation from the model can be used to estimate the parameters, but there's a minimum set that you need, which is these. And you can see that because there's a one-to-many relationship between the different parameters and the observations, that tightly constrains the wriggle room of what parameters can fit these set of observations. So it's a really powerful setup. You can actually estimate the parameters of this rather sophisticated model. And there's a process that's been set up to do this called the Add My Pet project. It's set up in MATLAB. There's a PLOS computational biology paper describing the whole thing. And it's been done for over 6,000 species of animal now. And all that information, all the parameters and all the code can be found on this website, the Add My Pet project. And when you go into there, you go to the species list, you'll see all the species. And skipped a slide. So this is the list of the species, and on the right here, all of the observations that were needed, all the functional trait data that was needed to estimate the parameters of the model. But the trait data is sitting inside MATLAB files, so it's not in a database. It's in code, and so you can see this is an example of the My Data file for a fish, and you can see it's got age at birth here at 47 days, and the temperature associated with that, the metadata temperature was 4 degrees. So there's a huge amount of information. All the metadata and everything is sitting in there, but it's not very accessible. So what Nat Briscoe and Chris Wild and I have been doing is writing code to pull all that information out of these MATLAB files and putting them into a database, and then seeing what we've got, what sort of coverage do we have, and what sort of patterns do we have. And it's a really powerful database because all of the numbers that have gone into it have been used to estimate parameters, which weeds out errors in the observations. And it's very complete in that there's lots of, every species has at least that bare minimum of observations that I showed you before. And when we look in at what we've got in this database, you can here's some of the numbers. The most common thing we have in the database is the ultimate wet weight, that's WWI, over 4,000 observations. But then there's over 1,600 observations of age at birth or incubation time, each with a temperature. And so we can then play around with that data and see how it looks compared to other databases. Here's a couple of plots of the age at birth corrected to a constant temperature of 20 degrees and the weight of the hatchling. And in the light blue in both cases are the data in that database, and then the other colors are data in other databases. But then the question arises, how do we organize all of this? And that's where traits.build comes to the rescue. So this is a setup that's relatively recent. This paper that was led by Lizzie Wenck and Dan Folster explains the whole process, and it's built off the AusTraits approach, and it's highly standardized in connecting with Darwin Core, the Ecological Trait Data Standard, the Extensible Observation Ontology, and FAIR principles. And it has a really nice setup for organizing in a relational sense your trait data and the associated metadata and the taxonomy. So it's actually beautifully receives all of the data that's come out of this estimation process that's sitting in those files. So we're pulling it all out, putting it in a database, but then ultimately, instead of putting the data hardwired into the MATLAB files in the future, we want people to put it into a database, and then we pull from the database to estimate the parameters. But then what's really cool about this is that once you have a dynamic energy budget model of an organism, you can get all sorts of observations out that of essentially estimates of traits that you didn't actually put in. This is a bit like reanalysis weather data, like the NCEP or the ERA5 data sets, if you're familiar with those, which are from a dynamical systems model of the climate system, but calibrated on observations of the weather. And so you get output from the model once it's calibrated without any gaps, because it's the output of a model. And it's the same with the Deb theory and all the things that can come out of there, even though you haven't put all those observations in the first place. So for example, a good example is metabolic rate, often measured as oxygen consumption rate, but usually not used in the estimation procedure. Like very few observations have this in the estimation procedure. But if we have the parameters estimated, we can produce the metabolic rate as an output. And so what you can see here in gray, the metabolic rate plot points from the database against mass. And then in orange, you can see data from a couple of different databases. So it's really powerful. And what we want ultimately, or what we can do, given that we have a dynamic energy budget model, of course, is link that to environmental conditions and predict trajectories of growth and reproduction and survival in nature under different climate scenarios and different management scenarios and so on. And that's what the niche mapper package is all about. So we're wanting to develop these databases, of course, so that we can have the parameters for these models, but it, as a side effect, in a way, from what we're trying to do here, we end up with these really tightly described and databases of traits. And we can then use this to ask questions about how species responses will vary depending on, in this case, 2 different scenarios of water availability for a lizard out in the desert. So in summary, I hope to have illustrated the power of collecting trait data in the context of a dynamical systems model of an organism, be that metabolism or heat exchange or behavior. And to have made you aware of this enormous trait data resource in the form of the AMP project, to have shown how modern trait data-based approaches are ready and waiting for this trait data and for parameterization of these kinds of models, and also the notion of reanalysis of functional trait data you can get from these kind of approaches. And of course, having this target set of data tells us what the research community needs to prioritize in the future, collecting trait data. Thank you very much.
Hey, so thanks everybody for coming along to the session today, and especially to all our wonderful speakers. I know we're short on time for questions. In the program, there was a panel discussion. That was a bit of an error, but I am going to ask if all of our speakers from today can just hang around up the front, and if you have a question, please come and find them. If you could just join me in thanking everybody for the presentations. Thanks very much. Ooh, for those of you yesterday, we do have a discussion session on Friday at 2 p.m, a fireside chat, like a more interactive discussion about the role of modeling in the global biodiversity framework. And we would love to see you all there. Please come along.
Vacation.